right, let's play this thing here. So what does it say? This is a nice cell. When we look at this cell, what do we see? We see a nice cell membrane outlining the edges. We see a nice nucleus inside. And then we see all these other things in here, all these other things. Mostly this is all cell fluid. A lot of times when you see the word cyto, like down here, I see cytology. This root word cyte means cell. So the fluid of the cell is called cytoplasm. All right, so cyte usually means cell. Cytology, ology is the study of, and cyte is usually the cell. So the human body contains trillions of cells, but these cells, even though they're genetically identical to each other, all the trillions of cells are genetically identical to each other. They can differ in shape and in function, because even though every cell has the complete book of DNA, they're not all paying attention to the complete book of DNA. Some of them are just expressing certain genes that allow them to take their specific form and allow them to perform their specific functions. All right, so a couple hundred years ago, some old dead guys came up with this thing called the scientific theory, and the cell theory. And how did they come about these ideas? Well, they needed to invent the microscope first, really, because really nobody knew what cells were before the invention of the microscope. So in 1600 and something, this guy, Robert Hooke, came up with the microscope, and then these ideas started to come from that. Number one being that all living things come from cells, right? That's the first part of the cell theory. It was first realized that all plants were derived from cells or composed of cells. And that came from a guy, his name was Schleiden. And there was another guy somewhere else in the world called Schwann. His name was Schwann. And he figured out that also all animals were composed of cells. And then we kind of took those two ideas, lumped them together and said, hey, listen, all living things are made of cells. Where do cells come from? Well, cells can only come from other cells, right? That's the second part of the cell theory. Cells contain hereditary material. So in the nucleus of the cell, there's DNA, and that DNA is copied every time the cell divides. So a copy of the DNA is then passed as the cell splits to daughter cells. All right. And cells are the basic unit of structure and function in living things. So we can't strip things down to smaller units than cells and consider those things living things, right? So if we have a cell and we take the nucleus out and put the nucleus in a dish, well, the nucleus by itself is not a living thing. If we take a cell and remove the mitochondria and put the mitochondria in a dish, well, the mitochondria alone in a dish is not a living thing. However, when we take the cell, complete cell with the nucleus, mitochondria, ribosomes, all the organelles collectively together, well, we can consider that one cell a living thing. All right, so the composition of cells is quite similar. We talked about that a little bit last week, the chemical composition of living things, organic molecules. That means that we see things like proteins, lipids, nucleic acids, carbohydrates when we look at how they're made up, right? So on the outside of the cell, we have this structure called the cell membrane. Sometimes the textbooks call it the plasma membrane. But anyway, it creates a barrier. And this barrier is what separates the inside of the cell from the outside of the cell. We looked at the structure of the cell membrane last week when we were looking at phospholipids. You remember the hydrophilic phosphate heads being attracted to the water, where the two lipid tails were repelling the water. So sometimes the cell membrane is referred to as the phospholipid bilayer, because we have those two layers of phospholipids making up its structure. The fluid portion of the cell is called cytoplasm, sometimes called cytosol. And we find cytoplasm between the nucleus and the cell membrane. So this is the fluid that completely fills the cells. And this is where you're going to find the rest of the organelles floating around. So in this picture, all this blue stuff is cytoplasm. All right. So in this red thing in the middle here is is the nucleus. So everything between the cell membrane, which is the outer membrane all the way around the outside, and this nucleus, this is all cytoplasm. You can see all these other structures are floating in it. The simplest forms of cells, and these we see these like in bacteria. Bacteria are prokaryotic cells. These are very simple cells, very basic cells. The only prokaryotic cells that we see today are bacteria. All right. So these life forms were, it's believed that they're the first life forms to be present on Earth. And what makes prokaryotic cells different from eukaryotic cells? Well, eukaryotic cells are the cells that we have in our bodies, right? And they're the cells that we find in plants, in protists, all the other kingdoms, right? So the only kingdom that contains prokaryotic cells is the bacteria kingdom, the eubacteria and the archaebacteria kingdom. And the difference is that they don't have a nucleus. They don't have any membrane-bound organelles. Even though they have DNA, their DNA is not stored in an actual nucleus with a nuclear membrane. It's just kind of floating around in the cytoplasm. They do contain ribosomes, however, because like all cells in the world, they have to produce proteins, and ribosomes are the part of the cells that make proteins. So they have to have ribosomes. But other than that, they don't have much. They have DNA, they have cytoplasm, they have a cell membrane, some of them have a cell wall, but they do not have an organelle. So where did eukaryotic cells come from? Well, it's believed that eukaryotic cells evolved from prokaryotic cells. And the theory is called the endosymbiosis theory. And what that is that theory is kind of explained as there were some prokaryotic cells kind of hanging out, and they engulfed each other. And instead of engulfing each other, eating each other, and then breaking each other down, like the stomach would break down your food, the larger cells realized that there was value to the smaller engulfed cells. And what happened is the larger cells became hosts for the engulfed cells. So the engulfed cells survived inside these host cells, and they developed a mutualistic symbiotic relationship where the smaller engulfed cell now had a safe space to live, and it was getting food by sitting in the cytoplasm, and maybe the smaller engulfed cell was giving something to the larger host cell. Maybe it was providing it with some type of chemical energy. So anyway, they evolved some symbiotic relationship. In fact, when we look at the DNA inside the nucleus of all of your cells, there's different DNA found in these other organelles in here. Like the mitochondria, for example, has its own separate DNA. So that is just providing evidence to this endosymbiotic theory that perhaps this came from somewhere else and has just kind of stuck around. So what's the difference between plant cells and animal cells? Well, plant cells have most of the same organelles as animal cells, but they have a couple extra ones that animal cells do not have. The cell wall is one of them. The cell wall is not a living portion of the cell. It's actually a non-living portion of the cell. It's made up of a really strong carbohydrate. We saw this in the notes last week. This carbohydrate is called cellulose, and it's a big, strong polysaccharide. It's so strong that we do not have the ability to break it down in our digestive systems. Another difference is in plant cells, instead of smaller vacuoles for storage, there's a large central vacuole that stores water. And when the plant cells fill their large central vacuoles with water, what it does is it puts pressure up against the cell wall. And when we have millions of cells that are doing that in a plant, filling their cell Their central vacuoles with water, putting pressure on the cell wall. It makes the plant as a whole stronger. It creates pressure inside there called turgor, and that pressure makes the plant stronger. In fact, you might see as a plant when it loses water, it will lose the pressure inside its cells and the plant will wilt, right? It'll lean over to the side. And how do we get it to come back? Well, we got to put more water in the soil. That water will then get absorbed by the roots and fill those cells again, putting more pressure on the cell wall, and then the plant stands up straight again. Plant cells also have structures called chloroplasts. And of course, this is the site of photosynthesis. So plants have the ability to make their own food from the sunlight. The cell membrane is made up of phospholipids. We saw that last year. Embedded in the phospholipids, there's these other structures, sometimes cholesterol structures, cholesterol-based structures, and proteins. But the phospholipids are a double layer, right? And the proteins and lipids associated with the cell membrane have these sugars that are attached to their surfaces. We call those glycoproteins or glycolipids. And a lot of times the glycoproteins that we see some here and glycolipids are involved in cell-to-cell communication, right? So here we see the phospholipid bilayer, the phosphate head, the lipid tails, phosphate head, lipid tails. The tails face each other. The tails are hydrophobic. The heads are hydrophilic. So there'll be water on this side of the cell, and there's water on that side of the cell. This is outside the cell. This would be inside the cell because it says extracellular fluid out here. So we know this is outside the cell. And then we have these channel proteins, right? Because due to the fact that this is a hydrophobic environment in here, a lot of things won't be able to cross through here. Like water. This area repels water. Water won't be able to come through here. But water will have its own specific channel protein that will allow it to go from the inside of the cell to the outside of the cell. All right, so a lot of substances, glucose, water, salts, have to pass through these channel proteins that are embedded in the cell membrane in order to go from outside the cell to inside or inside the cell to outside. So the glycoproteins and the glycolipids form what's called the glycocalus. And what this does, it's kind of like a fingerprint for the cell. It kind of advertises the cell as being self or being non-self, right? Because if you could imagine, you have white blood cells that are traveling around, constantly feeling your cells. And if they feel a glycocalyx that is foreign, they're going to react to that. They're going to create an immune response. But your glycocalyx are not foreign. They're part of your body, that you make them. They're specific to you, and your white blood cells know that. All right. Your white blood cells have identifying proteins on them, and that's what gives us our blood types, right? So these are just markers that inform the white blood cells that, hey, these cells, these belong to us. Don't attack these ones. Look for foreign invaders. Look for glycocalyx that don't belong. React to those. Well, this is a good point too. When we're looking to match, so let's say, for example, somebody needs a kidney. You don't just take any random kidney and put it into somebody. You try to find a kidney that matches its tissue type. Well, what were we looking at? We're looking at the cells of the kidney, specifically the glycoproteins and glycolipids that make up the glycocalyx, and we're trying to find as close a match as possible. All right. The cell membrane has a fluid mosaic structure. What does that mean? That just means that it has the ability, these phosphate heads and lipid tails, they do have the ability to come apart and allow things to pass through, but then they come back together again. What causes them to come back together again? Well, that happens as a result of the hydrophilic properties of the heads and hydrophobic properties of the tails. So it's not a solid structure. It's more of like a fluid structure, right? It's constantly moving and swirling around, similar to olive oil and water, right? In the lab, we talked about in order for osmosis to happen, you have to have a semi-permeable membrane. Semi-permeable membranes that are membranes that allow certain things to pass, but not everything. So when it comes to our cells are considered semi-permeable because it means that nutrients are allowed to enter the cell, waste products are allowed to exit the cell, and water, of course, could go back and forth, and gases, right? These kinds of things, but not everything. Some things are too large, right? But that's why we have a digestive system. We want to break down these nutrients. We could get things to cross the membrane in two ways: passive transport and active transport. Passive transport is diffusion. Okay, active transport is the opposite of diffusion. Let me go here for a second. Let's go to here. This way we can kind of review this. When we're looking at diffusion, remember diffusion is a form of passive transport. So passive transport means that there is no energy required, right? So what does that mean? That means, let's say we have a cell here, right? And there's molecules inside the cell, but there's a higher concentration of molecules inside the cell compared to outside the cell. Well, these molecules are going to move from an area of high concentration inside the cell to an area of low concentration. And this will happen without the use of energy. Active transport, on the other hand, is, it's kind of the opposite. So let's say we have a cell and there are more molecules inside compared to the outside. So this is active transport. Remember, active transport, this can happen in our bodies. Of course, it happens all the time. But this requires energy. Requires ATP, right? That's our energy molecule. Let's say we want to get to, these cells want to move molecules from an area of low concentration to an area of high concentration. That could certainly happen, but that's going to require the use of a pump, right? So like a sodium potassium pump that would be embedded in the cell membrane. Of course, that's going to use energy. We need energy to do that. So there's two forms that the body would use to move substances across cells. This one does not require energy. So this is passive transport. This would be diffusion. This would be osmosis. And then we have the opposite, which is active transport. All right. So when we're going from low to high, we're going up the concentration gradient. This is against the concentration gradient. So this is kind of like going uphill. Like if you're on your bicycle, you would have to apply energy to do this, right? But if we're going from high to low, this is going down the concentration gradient. If you're on your bicycle, this would be like going downhill. You would not need to apply energy. Okay. So in this situation here, we have a semi-permeable membrane going right down the middle of a beaker here. You can see it. And on this side, we have an area of high concentration of molecules. And they're going to want to cross this semi-permeable membrane and go over to the side. And that's what's happening here. They're filling in that space. And that's going to continue to happen until both sides become equal, right? And then we're going to have molecules going back and forth. So this, we call this an equilibrium right here. So this is diffusion. Remember, the diffusion of water is called osmosis, when water moves across the semi-permeable membrane from an area of high concentration to low concentration. We saw osmosis happening when we put our red blood cells in hypotonic solutions as well as hypertonic solutions. Because when we put our red blood cells in a hypotonic solution, the higher concentration of water is out here. The lower concentration of water is inside the cell. That's going to force water in, causing the cells to burst. When we put it in a hypertonic solution, there's more salt in this water, so there's a higher concentration of water inside the cell compared to outside the cell, and that's what causes the water to leave the cell. So usually a good way to remember this is water will go where the salts are. If there's more salt inside the cell, then the water is going to go inside the cell. If there's more salt in the water, then the water is going to leave the cell and go into the salt. All right, so that's how we end up with these changes in shape. This is called hemolysis. Lysis means to break, right? The cells are broke. Hemo means blood cells, right? Hemoglobin, hemolysis. Crenated cells are the ones that shrivel up. We saw these on the microscope. And these did not change shape because the concentration of water on this side was the same, which was what? 0.9, right? 0.9 salt. Same thing on the inside, 0.9% salt. So that's not going to change anything. That's an isotonic solution. So facilitated diffusion happens like before when I said, when we have these phospholipids making up the cell membrane, and then we got the tails here. These are the hydrophobic tails. Facilitated diffusion happens when we have to use a channel protein to move a particular substance from one end of the cell to the other end. So it's still moving from high concentrations to low. It's still diffusion. But real diffusion is when the substances could cross right through the cell membrane like this. If we have to use a channel protein that's embedded in the cell membrane, like this red one, it's still diffusion, but we're kind of helping it along. So we call that facilitated diffusion. All right. Here we see a good picture of facilitated diffusion. These solutes out here, they're not going to be able to get through this way. So they have to go through the channel protein to get to the other side. It's still going from an area of high concentration, these solutes, to an area of low concentration. So it's still diffusion, but it's being facilitated by this protein right here. Or active transport. Again, active transport is the opposite. We're moving molecules against the concentration gradient. So we have to use a pump. In this case, we use sodium-potassium pumps. And of course, we have to use energy in the form of ATP. And that's how we get substances to cross the membrane. We use an actual pump. And this uses energy. All right, so what do we use active transport for? Well, when we want to get all the nutrients from the small intestines into the blood, right? When we fill the intestines with nutrients, they're in a high concentration. But once most of them are removed, we want to pump whatever's left in there. Into the blood, whatever nutrients are left in the small intestine, we want to get them all into the blood, right? Another example would be, let's say we have in your bones, right? There's blood in your bones. So let's draw a blood vessel here. My red isn't working. Here we go. Let's say there's a blood vessel here, and there's some calcium. let's say the blood calcium levels are elevated, but your bones are made of calcium, right? So this is where calcium is stored. So there's always going to be a high concentration of calcium inside. But let's say you have an elevated level of calcium and the body wants to store more calcium in the bone. So this would be a low concentration of calcium in the blood. You could still pump this calcium out of the blood and into the bony matrix here. And you would need to use pumps to do that. And this would be an example of active transport. Sometimes the ribosomes make proteins, and the proteins get packaged up by the Golgi here. When they get packaged up, these little packages called vesicles would then migrate to the cell membrane and release these proteins. This could be something like a hormone being released from a cell, right? This is called exocytosis. When the protein product is sent to the Golgi, packaged up, and this secretory vesicle then migrates and fuses with the cell membrane to release that product. That's called exocytosis. Exo means out. Remember, cyte means cell, so it's going out of the cell. On the other hand, if there's something already in a vesicle from somewhere else approaching the cell, this vesicle can then combine with the cell membrane, and these products in here can be contained in another vesicle, brought in, though, they're going the other way. So this would be called endocytosis. Endo means in. So we could bring products in this way, or we could get products out of the cell that way, both using secretory vesicles. Let's say that this is a food nutrient being brought to the cell. The cell will encase it in a vesicle, and then we can meet up with a lysosome. Lysosomes have digestive enzymes in them. So the lysosome will meet up with this. The digestive enzymes will break that food nutrient down, and then later on, those nutrients can be released inside the cell. All right, so this is the picture of an animal cell, typical animal cell. And we talked about the cytoplasm. We talked about cell membrane. And that's really about it so far. The cytoskeleton is provided with support from this structure called the cytoskeleton. It's kind of like a cellular scaffold. And it's used for not only suspending organelles, but helping to move organelles around. All right, so the cytoskeleton gives plasticity or fluidity to the cells. It allows the cells to change shape and move organelles around. The cytoskeleton is made up of a couple different things: microfilaments, intermediate filaments. These are similar to cables. These are like long, skinny filaments, and then microtubules. We see microtubules appearing during mitosis, reproduction. The flagella is a whip-like tail. Sperm cells have a flagella. So if I was to draw a sperm cell, I would draw a head and then a midpiece, and then this long whip-like tail, right? The whip-like tail kind of propels the sperm cell. So this is how the cells move around. Not all cells have these. Some cells have cilia. There's some protists that have these little hair-like structures on the side called cilia. And as the cilia move, it kind of propels the cell like oars in a boat. Your respiratory tract has ciliated cells in it. And what happens is when you breathe in dust and other undesirables, they get trapped in the mucus in your respiratory tract, and then the cilia kind of brush them back up where they came from. The ER, endoplasmic reticulum. So reticulum means network. Endoplasmic means in the plasma. So inside the cytoplasm of the cell, there's a network, right? It's kind of like a network of tubes or canals. And we call this the ER. And we have two types of ER. We have ER that is considered rough, and it's this ER that has ribosomes on the surface. Ribosomes make proteins. That's what they do. So if the ribosomes are sitting on top of the ER, it gives that ER a rough looking appearance. So we call that rough ER. If ribosomes are not sitting on the ER, well, that section of the ER is not really going to be involved in making proteins. That section of the ER is probably going to be involved in making lipids, fatty acids, hormones. Because the ribosomes aren't sitting on there, that ER has more of a smooth appearance. So we would refer to that as smooth ER. However, both types produce vesicles that are filled with a product ready for the next step. And usually the next step is going to involve sending this product to the Golgi so it can be packaged and then sent out for exocytosis. So the Golgi is like the packaging center. This is like the UPS of the cell, right? This is where the cell products, let's say the ribosomes made a protein, that the ribosomes hand it off to the endoplasmic reticulum. The endoplasmic reticulum is a series of canals where the protein is finishing being processed. Well, once it gets to the Golgi, it's going to be packaged. This protein will be packaged in a vesicle, and that vesicle can then, here we see, this is what the Golgi looks like here. So the ER would be on this side, and the proteins that the ER are receiving from the ribosomes would go to the Golgi, and then the Golgi would package them up in these vesicles, and then these vesicles would migrate to the cell membrane where they would be released from the cell. Lysosomes, remember, lyse means to break. We saw hemolyzed cells, the cells that took in too much water, the cells that were sitting in a hypotonic environment, they hemolyzed, right? So lysosome, lyse means to break. These are digestive centers. These have digestive enzymes. So what these cells do, look in a couple slides before, we could see these are membrane-bound organelles. They have a big membrane, and they contain these enzymes. And what they could do is they could link up to another vesicle and break whatever's inside that vesicle down. Well, what could we break down? Well, let's say there's a piece of bacteria that the cell engulfed, or let's say there's food nutrient. So we could break those things down, and those items could get recycled. Now, the nucleus is very important. A lot of times it's the largest organelle. You can see it very easily. Here's the nucleus here, and you can see it has its own membrane. And in the membrane, there's pores. There's nuclear pores. The nuclear pores are very important because it's in the nucleolus here that ribosomes are made, and ribosomes would have to leave. Also, it's in here that we have the DNA, right? The DNA never leaves, but sometimes we make copies of the DNA in the form of RNA. And of course, the RNA would have to leave through these little pores, the nuclear pores. Mitochondria are very important. I don't know, in high school, you might have learned that the mitochondria is the powerhouse of the cell. What does that mean? It means it's the site of cellular respiration, right? This is the site of cell respiration. Well, what's cell respiration? Well, this is a process that our cells use to provide us with energy. So we have to provide the cells with a couple of things in order to get energy. We have to breathe in oxygen. We have to eat food and get nutrients like glucose. Okay, we combine these two things, and the cell, as a result, is going to make energy, ATP, for us that we can use. Probably a bad color choice. ATP. But it's also going to produce some waste products. These waste products, believe it or not, are going to be carbon dioxide. And also another waste product of this is going to be water, right? A lot of times you don't think of water as a waste product, but when it comes to cell respiration and making ATP, it is. Your bodies are constantly making water. This is a double membraned. Has an inner membrane. You can see that here, these inner folds, and then it has an outer membrane. And this is cell respiration here, which I'm not gonna, you don't need to know all that. So how do cells communicate? Well, we use hormones a lot of times, right? And we could use hormones in a couple different ways. This is a secreting cell. Right? So this cell here is producing a hormone and releasing it. Right? Some hormones, when released, get picked up by the blood and they could travel throughout the entire body. So they could have an effect or communicate with cells that are on the completely opposite end of the body, really far away. These are called circulating hormones because they get picked up by the blood, they travel in the blood, they leave the bloodstream and communicate with target cells that are far away. Local hormones could communicate with target cells, but don't need to get picked up by the blood. They could just kind of travel to nearby cells by traveling through extracellular fluids. And then we have gap junctions, which this is a cell that's producing a hormone. This is another cell that's producing a hormone. And these chemical signals can be shared through these tight gap junctions between the cells. All right, so these cells could communicate because they're neighboring cells. There's little tunnels here that allow these chemical messages to go back and forth. So there's three different ways: circulating through the circulatory system, local hormones, and then gap junctions. And this is just kind of saying what I already just said. All right. And this concludes the lecture. Pretty simple. I mean, I would know the parts of the cell theory. I would definitely know the difference between diffusion and osmosis, passive transport and active transport. And then I would definitely know, make a list of the organelles and their functions. All right. Any questions? Give me an email and I'll be happy to help you out. Okay. All right. Look for the lab. Fetal pig coming next.